Ahora vamos a tener al doctor Mike Martin de la Universidad NTNU en Noruega. Su investigación consiste principalmente en analizar secuencias del genoma para reconstruir e interpretar historias evolutivas de plantas, animales y sus comunidades microbianas asociadas. Él se interesa particularmente en el uso de ADN antiguo, ADN degradado, derivado de colecciones de historia natural, para comprender las distribuciones actuales de la biodiversidad mirando directamente al pasado. Y me complace mucho que el doctor Mike Martin haya aceptado estar hoy día con nosotros. Está siendo en este momento supervisor del trabajo de tesis de maestría de uno de mis estudiantes, Renato Latorre, con quien todos estamos muy contentos. Bienvenido, Mike.
Gracias. Thank you, Gisela, for the invitation, and also thank you to Jorge Ramirez, who I know is somewhere in the audience, and it was so nice to invite me here to speak to all of you. And I am saying hello from the other side of the world, where it is feels like the end of the day, quite dark now. We have heavy snow coming down today. So I'm dreaming of being there in Peru, where I know you are having probably some sunny weather today. Yes, so thank you. I'm today going to talk about, I'm going to introduce you to the world of plant genomics, and I will hope to inspire you that this is an exciting and interesting field to be involved with, and maybe some of you will be working on master projects somewhat related. I don't know, but I find it an exciting area, and I'm going to be happy to tell you about a few different projects that are all revolving around the use of museum specimens for genomic analysis of plants and also the microbes on the plants. So really the work I want to talk about today is taking place inside herbarium collections. So these are parts of natural history museums where you find the dried and pressed plants. These collections are often quite large and very well curated and have been historically used by taxonomists for systematic studies, as well as sort of reference libraries for the morphology of plants, and also a bit of information about when plants were flowering at times in the past. Color variation, all kinds of things can be extracted from these really well curated collections. And these collections are actually quite extensive. There are estimated to be around 3,000 herbarium collections around the world. And there's a total of something like 350 to 400 million plant specimens contained within these collections. And we know that these collections started to be put together as in Europe, at least as early as the late 1500s. So extremely early time in science history. And the number of specimens preserved in these dried collections has increased dramatically over time. And we sort of are peaking in the last 100 years with huge numbers of specimens that are available for our research. And that's an interesting time for us to be looking back to look at these plants, because this is, of course, when humans have been heavily disturbing global ecosystems. So what kind of stories can we tell, and what kind of questions can we answer by looking into the past with these herbarium collections? And I would like to introduce the idea that these are not just libraries, reference libraries for plant taxonomists and for botanists, but in fact, they can be considered genomic resources. these collections. We have found now that the DNA content in the herbarium collections is actually quite well preserved. I'll tell you more about that in a bit. So the idea here is we take a small piece of whatever bit of plant has been dried on the paper. We put it into our laboratories, extract DNA, prepare genomic libraries, sequence them, and then start to infer exciting things. Especially when we have really nice and plentiful collections for some plants that are really within the last 150 years. We have a lot of material to work with, depending on which part of the world you're looking at and how well studied that plant, or how well collected that plant has been. So what has been happening with herbarium genomic analysis so far? Well, there's actually been quite a lot of work going back the past perhaps 15 years. Really about 10 years has been really a lot of studies coming out. And what we know basically is that the decay of the DNA, the degradation of the DNA that's in these plant specimens is happening very quickly. It's actually happening more quickly than, for example, bones that are being preserved also in natural history collection from animals. It has been found that the plant DNA is decaying about 6 times faster. So that's interesting. Perhaps says something about the way that the plants were preserved, often using heat to dry them before they were put on the paper. Or perhaps something else about the cell wall or the cell content of the plant material. We also know that something called DNA damage accumulates in these plant specimens. And this is something that has been investigated with ancient genomic data. It was first sort of discovered in the ancient human genomes. And what we mean by DNA damage is that there are chemical modifications that happen at the ends of the DNA fragments that are directly associated with hydrolysis of cytosine bases at the very ends of the fragments. So the fragments are first very long due to heat and other chemical effects and age. The fragments are torn, and then at these edges, we start to see these lesions in the DNA. And these lesions can be recognized when we build libraries and sequence the DNA as mismatches that are more frequent at the ends of the DNA molecules and then becoming less frequent as we go toward the center of these short molecules. And they're basically mismatches to the reference genome that the DNA has been mapped to. So we use this pattern to authenticate ancient DNA and to identify when we may have been amplifying modern DNA as opposed to authentic ancient DNA from an ancient or very old specimen. So we find DNA damage in these herbarium specimens, which correlates with age or the year of collection. You can see that here. Also the degradation of you can see that here, the damage fraction. And the length of the fragments that we can recover from these specimens is also highly correlated with the amount of time since they have been collected. So over time, the DNA gets shorter and shorter and shorter and more damaged. But the nice thing that we have discovered looking at historic herbarium specimens is that they actually have quite a high proportion of plant DNA left over. And this can be a big problem with museum collections, that over time, what starts off with a specimen with 100% DNA from the host organism is these specimens are colonized by bacteria and other microbes, and those microbes can become a very large proportion of the total DNA available, which makes them actually very difficult and very expensive to sequence, because we have to sequence a proportionally higher amount of DNA to recover the entire host genome. But this is not such an issue with plants, which is a good thing. Okay, and I mentioned that these studies have become more and more common, and this is this is there's quite a few review articles out there about recovering ancient DNA from herbarium collections. There was a recent review in Science. Actually, I think it came out this year. So if you want to get into this field, you can take a look at some of those recent reviews, and maybe that's a good starting point to find where your study might fit in. The kind of biggest veins of research with historic herbarium genomics have been coming from a really good group that's situated in London, the Hernan Barbano group. And he has been working on historic potato specimens, so the history of the introduction of potato to Europe and the adaptation of the potato genome to European climate, which has actually been a really interesting story to follow, as well as the introduction of Arabidopsis lineages or Arabidopsis populations from Europe into North America. And using those herbarium specimens to sort of track the rate of mutation accumulation over time. My own work has started in herbarium genomics, started with investigating the introduction of the potato pathogen called Phytophthora infestans, which is an oomycete, something like a fungus, to European crops, potato crops. So when this pathogen was introduced around the year 1845, actually 1843, in the UK and Ireland, it caused a massive die-off of potato plants. It was an epidemic, and an epidemic that spread all across Europe and caused the Irish potato famine and the exodus of Irish people and the starvation of millions of Irish people and their distribution around the world almost. So what we did was to use historic herbarium specimens of the potato plants collected from 1845 and to sequence the lesions where the pathogen had made an infection on some of the plants, and to recover the genome of the pathogen, and then to relate that to the potato genome, which is work that I'm proud of. And since then, actually, plant pathogens have become a big part of what's being done in herbarium genomic research. There's several studies on the way out where we have used these herbarium specimens, which, as I mentioned, are very plentiful, to detect and track like the first introductions and the earliest introductions of particular plant pathogens, and to put them into evolutionary analyses so we can find out exactly which strain was introduced and try to track the origins and study how we might be able to predict these introductions in the future in order to increase global food security. We also used, back in 2014, we used some of the first herbarium genomic data to characterize the metagenomic community that was found on these herbarium specimens. Actually, in this case, they were potato leaves. And that was an exciting kind of early result showing that in each herbarium specimen, on each leaf that we analyzed, we basically looked at all the sequence data that didn't map to the potato reference genome, but we looked at everything else, all the unmapped information, which is normally just discarded. But when we put those sequences through a taxonomic identification pipeline, we were able to recognize that each had a characteristic microbial signal. You could call it a metagenomic or microbial community with different bacterial taxa represented in different proportions. So although we didn't investigate that any further, this is the first indication that this sort of work can be done. And I'll return to this kind of work because this has really become our focus in recent years. But really our main focus these days with herbarium genomics have been to investigate the process of invasion. So the process of plant invasion. we'd like to know, how do plants become invasive? Which plants become invasive, and what are the genetic mechanisms, the evolutionary, what's the evolutionary basis of these invasions? So rapid adaptation, from one hypothesis for how some plants can so easily adapt to a novel range upon introduction is that they can rapidly adapt from the variants that already exist in the population in the native range. If any of those variants are particularly useful in the introduced range, then those can rapidly rise to high frequency. And there has been suggested that this process is happening in parallel. Whenever a species is or plant species is introduced to multiple introduced locations, the hypothesis is that the same genes can be used in parallel in multiple locations to produce the same invasive phenotypes. There has also been suggestions in the past that admixture in the native range between formerly differentiated populations can create these sort of hybrid plants that are especially well prepared to evolve in the native range, in the introduced range, because they have high levels of genetic diversity. Okay, so sort of a hybrid vigor hypothesis. And the third hypothesis about why particular species can become invasive upon introduction to a new range is that they are basically being released from enemies, either herbivores in the case of plants, plant animals that are eating the plants in their native range, or co-evolved pathogens that if they can manage to escape from them in a new land, a new range, they can then use the resources they had formerly devoted to fighting the enemy for growing faster, growing bigger, reproducing more, etc. So the nice thing about herbarium specimens is that they're often highly abundant and often really well characterized by metadata, like a location and a year, and even a time of year, even down to the day. You sometimes know when the plant was collected, and then it's kind of frozen in time on the sheet, dried in its natural state. So this allows us to go back and think of experiments that we could conduct. These introductions can be thought of as evolutionary experiments, where the species is introduced to a new place, and we can watch over time, if we have the specimens available, how the evolution happened. So the case study for our invasion genomics work has been common ragweed in genus Ambrosia artemisifolia. This is a plant related to sunflowers, Helianthus from North America. It's a North American native and a weed that's highly invasive in most of the places where it has been introduced, especially in Europe. Incidentally, the pollen from this plant is produced in very large amounts, and it is all pollen that drifts through the air and the wind and causes what's called hay fever. This is seasonal allergies in North America and in Europe. It's actually a big economic burden. People are treating this effect from pollen from this plant and other plants too with medications. So it's a pest. It also takes away resources from farmland, farmed crops whenever it shows up, because it will sometimes become very abundant on a farmland and kind of dominate the crops unless it's controlled. The plant was introduced to Europe in the 1800s, and then since then it actually has been introduced all over the Earth except on Antarctica. It's not invasive everywhere, but it is pest and is highly invasive in Europe. And we actually know from pollen analysis of lake sediments in North America that ragweed has had a long history of being associated with the local climate. We know that over the past 20,000 years, the distribution of ragweed, as told by its pollen in the lake sediments, has been changing in response to the glacial retreat in North America. And in particular, around the time of European colonization of North America, ragweed had a kind of explosion in abundance. And this horizon, or this time in the sediment core history in North America, is called the ragweed horizon. It's the point where ragweed went from ambrosia pollen went from around 1% of the total abundance of pollen in the sediments to sometimes up to 90%. And it's in each location where these lake cores have been taken, it's associated quite well with the arrival of European agriculture in that region. So Europeans cut the forest and converted the forest to farmland, and ragweed really liked that and did well. We also know from previous work on ragweed that the plants that grow in Europe, the introduced range, that the plants and the populations that descended from those initial introductions in the 1800s and 1900s are larger than you find in the native range. They also have a larger investment of resources in reproduction, like more seeds, bigger seeds, more flowers, producing more pollen. And also, it has been found by some really good researchers that were investigating this plant that latitudinal clines have been introduced for things like flowering time and reproductive biomass. So basically, this shows that the genetic variants that were introduced to Europe, probably in a random way, initially rapidly experienced selection, so that you see that flowering time is now highly correlated with latitude in Europe. So that flowering time is earlier in the northern latitudes, where it's colder earlier in the year, and flowering time comes later in the southern latitudes. Okay, and this has been called a story of rapid adaptation. There was only 100 or 150 years for this process to happen. Nowadays, here you can see in red, ragweed is present in high abundances and all over the northern hemisphere and on the east coast of Australia, and in many places in Eastern Europe and also in France. You could consider the plant to be invasive, and they spend a lot of money trying to control the spread of the plant. Okay. So our interest was to investigate the genome of ragweed. And first of all, we would like to do that using, we need a reference genome in order to map our data and do some population genetics, which is kind of my interest. We initially started back in 2013 with a single plant that we sent off for building some highly contiguous library building in order to assemble the genome. And regrettably, we chose a very high heterozygosity individual, which caused a lot of downstream problems. And we basically had a very low quality genome. But the plant actually is highly heterozygous. So between 2 and 3 and 100 base pairs is heterozygous in one individual. It's like 20 to 30 times the amount of heterozygous sites that you find in a human genome, for example. So it's a difficult genome to put together. It's highly repetitive, but we were able to make something of the initial assembly, and we could predict around 35,000 genes producing protein products using an RNA-seq approach. And these days we have a much better assembly that I will be talking a bit more about later. But this initial study I want to tell you about was using this highly fragmented assembly. So I would like to tell you about it's a study that's very exciting to me, and it was about trying to track temporal changes over time in ragweed populations, both in North America, the native range, and in Europe, the introduced range, where we have the invasive populations. And this was almost entirely the work of Dr. Vanessa Beeker, who was my PhD student at the time, and this was basically her PhD project. So our strategy was to go into the herbarium collections. Here you can see Vanessa working with sampling one historic herbarium specimen. And we designed basically a sampling scheme in which we would try to cover the whole breadth of the native range, where the plant is abundant, and also the introduced range in Europe. And we would collect as many individuals as we could, as many as the curators of the museum collections would allow us to take. And then we would match it with my own field collected samples taken from all over North America on some giant road trips I did back when I was a graduate student. And in particular, we wanted to target places where in the historic period, we had enough specimens where we could call the ones that came in a close, small location to be a population. And we tried to match those locations where we had data from, 150, 100 years ago in one location. For example, these are the locations where we had populations. We wanted to match them with modern collections from the same place so that we could look at allele frequencies changing over time, evolution in the population at that location over 150 years. That was kind of the goal. And we did this with approximately 700 whole genome sequences from this plant. And the genome was not that big. It's about 1 gigabase, but it still was a lot of data. And the way that we could kind of afford to do that was to use these fancy methods for low depth sequencing, kind of shallow shotgun sequencing that allows you still to do population genomics. And we did that using genotype likelihood-based tools rather than called genotypes. So the software that, in particular, that allows this kind of work to happen in a doable way is called Angst. You can check it out. I highly recommend the software. Okay. And we worked in our ancient DNA lab that we have here at NTNU in Norway. So it's a highly controlled lab where the airflow is always from inside the lab to outwards so that no modern DNA can come in when the door is open. So that's called a positively pressurized environment. And we work with double gloves and a face mask and a hairnet and lab boots and a bodysuit, all kinds of stuff. So it's like a clean lab environment in order to minimize the modern DNA contamination. All right, so Vanessa did a huge amount of lab work and sequencing, building DNA libraries, and the first results that we were able to produce were looking at, first of all, just looking in the native range in North America, looking for changes in population structure over time. So that's what we wanted to know. Were there any big changes happening in the native range that could explain which kind of variants were introduced to the introduced range? This is about testing the hypothesis that admixture in the native range between formally differentiated populations is contributing to the success of the plant. So we put all of those genomes on a PCA and colored them by, or tried to color them by the locations where they had been collected. Okay, so this is all samples collected before 1940, going back to something like the early 1800s. And we see some general patterns, okay, that the samples from the northeast part of the US and this part of Canada are kind of mostly focused here on the PCA, The southeast U.S., the warm semi-tropical part of Florida is all coming here. And we also, the blue is blue here. And we have this western part of the U.S. Mostly focused here in the PCA. And what we found was that there was this region that had been highly mixed between there's basically genotypes from the west and from the east. And that was here. And this is directly a result of all the recent expansion of agriculture into the U.S. over that time period, the 1800s to the early 1900s. And this kind of had been predicted from past genetic work. So we weren't surprised to see that. When we look at the modern specimens that were all collected around the year 2010, we saw basically the same population genetic structure. So no big changes there. Okay. Then when we further added all of the European specimens to the PCA, we started to see some interesting stuff. So here we are looking only at the historical European specimens. So those collected in the 1800s all the way up to before 1940. And it looks like a total mess, in my opinion. So that's all these gray dots and triangles, and they kind of didn't have much structure. You can see that they all kind of focused on this inside this oval, right? So that means the Florida specimens weren't being brought to Europe at that time, which wasn't a surprise. So yeah, the Florida was excluded, but all this the genotypes were basically introduced from all across the northern part of the U.S. But then when we look at the modern specimens in Europe, well, we see a little bit some different stuff going on. When we look at when we divide up the populations according to their ancestry, we can choose how many different ancient populations are represented in mixtures in the introduced range. We saw that there were some populations that were basically looking very different from anything from the native range and anything from the historical European range. So they had experienced some kind of differentiation, and a lot of it kind of fast. So here we have 4 points where we identified these populations with very different allele frequencies than we had seen in Europe or in North America. So those are interesting to us. And you can see they kind of like show up on the extremes of the PCA. So then what we wanted to do was one of the most exciting kind of approaches that we do in my group, which is called FST scanning or genome scanning, where we take two populations and look at the frequencies of all the variants in the two populations, and we put them side by side and scan for the variants that are the most different. So you can basically take, in this case, historic Europe versus modern Europe, and we line up the genomes and look at the allele frequencies of every SNP polymorphism through the whole 1 gigabase of the genome. And we look for those that are not directly on the line here, which would be along the line would be like no change in allele frequency over time. We are interested in these little bits on the outsides that changed a lot in a short amount of time, 100 years. And we want to know which SNPs or which polymorphism those are and what genes they fall in. And then when we do that, we are able to kind of summarize all of these like extreme outlier SNPs. And we asked which types of genes are they in, because we know something about the function of each of these genes in the genome from studies that have been done on Arabidopsis and other model plants. And what we found is that many of the changes that have happened over the last 100 years in Europe, from the early introduction time to today, have been in genes that are related to salt tolerance and tolerance to dry conditions. Also genes related to flowering time, which excited us because this is related to these clines that we already know exist and which were already associated with rapid adaptation in Europe to the new climate in Europe, as well as the germination of seeds, so reproduction. And in particular, we were kind of excited to see that some of the genes, many of the genes that were rapidly changing over time were involved with defense against pathogens, defense against basically the immune system of the plant, genes involved with that. So that was cool. So the basic, what we could conclude from this study, and this is just a quick summary, was that we could confirm there had been rapid adaptation to the novel environment, and we could find the genes that were directly involved in that process. That the introductions to Europe had come from multiple sources all across the North America, and it's particularly the United States and Canada. And there were some hints that this enemy release hypothesis might help us to explain some of the success of this plant. Okay, but we didn't have enough data to go there yet. So Vanessa got her paper published, and she was very excited about that. And that was her PhD. And then we were really lucky to be involved in a collaboration with Kay Hodgins' group, Monash University in Australia, who took it even further with all of these herbarium specimens and like the 700 genomes that we had available, and did a study that really looked in detail with the climate adaptation. For that, we produced a new reference genome that was way, way better. This is just how much the technology has advanced in 10 years. Now we have a ragweed genome that's fully chromosome resolved. In fact, both haplotypes, because it's a diploid organism, both haplotypes were fully resolved into the 18 chromosomes that are present in the genome. It's really high quality with the number of genes. Here, the blue just means that all the genes you would expect to find, the highly conserved genes, were present in the genome, which means we got most of them, and we can be kind of confident that we're looking at the whole genome and not just pieces. So the next study was about investigating using information about what's known from actual phenotypes in these populations in Europe and North America. Basically, some previous work had been done collecting seeds from these populations and growing them in a common garden environment, and then tracking the traits of the plants and associating that with the genotypes. So we kind of already have this information about which genotypes are involved with which traits. And these are relevant traits like growth rate, growth size, flowering time, this kind of thing involved with the rapid adaptation. And then we did some analysis of associating the environmental variables with the specific alleles involved with adaptation. And most of this work was the result of Paul Batelet from Australia, from Kay Hodgins' group, who the only picture that exists of this guy on the internet is one of him skateboarding when he was younger. So he's like because he was a pro skateboarder before he became a plant scientist. So just quickly with some of the cool results from this study were if you scan some of those locations where we had the population data from like 150 years ago and today, in North America, we had 5 of these locations. We did the genome scanning and identified regions of the genome that were highly differentiated over time. And what we were finding was like basically large blocks of the genome, contiguous regions, like 5-10 megabases contiguous were changing over time. So we, of course, wanted to know what kind of genes were in there with more detail. And we had the same thing going on in Europe. And in fact, we had even seen before that some of those populations in Europe were like highly diverged from the first introduction. And here in particular, there's a good example in Berlin population on chromosome 2. There was a huge piece of the chromosome, like 10% of it, which is like 50 megabases of DNA, was like very changed in allele frequency over time. And what we kind of discovered was there are lots of these large contiguous regions of altered allele frequency. We called them haploblocks. And with quite a lot of investigation, we were able to identify that these were chromosomal inversions. You can see here where you're plotting haplotype 1 of chromosome 8 versus haplotype 2. You see, as you would expect, that they're kind of like 1-to-1 aligned, and then suddenly they're opposite aligned. So this is like the whole region has been inverted on one haplotype versus the other. And then it goes back to a linear relationship, and then a huge inversion, and then a linear relationship. So we found lots of those. There were something like over 20 of them that we detected, and a few of them we could like be very sure they were chromosomal inversions. So this was a bit surprising. We didn't, but it was because we had a really nice genome. We could do this kind of analysis. And what we found was that here's 4 cases where the chromosomal inversion, we could track it over the presence of it or the frequency of it over time. So what we found was that these chromosomal inversions, or haploblocks as we call them, were highly correlated with latitude in Europe and in North America, and also changed in frequency over time. So that's what we see here, where the green is the older specimens from the herbarium, and they're kind of grouped by time, and then the purple is the youngest ones. We could see that these haploblocks were changing in frequency, and the latitudinal cline was being established over time. Okay, now we're like seeing the direct evidence of that. This one here, called chromosome 16B, is shown here. It's basically a huge piece of chromosome 16. I believe it's this one here. And you can see it's like changing over time. And what we found was that the frequency of these chromosomal inversions was highly correlated in these 4 cases, at least, with these phenotypic characters like the height of the plants in that population, or the flowering time, or yes, you can see that. And so if it has 0 copies of this, if the population has 0 copies of this haploblock, then it has a particular 100-day flowering time. And if it has 2 copies, then that flowering time is reduced. So some of these climate adaptation loci, SNPs variants are inside these haploblocks. Yeah, so they're associated with about 26% of the variation that we could link to rapid climate adaptation. We saw huge frequency changes over time, which was really cool. And we found evidence that some of these haploblocks were being used in the same way in parallel adaptations, like in different populations in both continents. You can think of these, maybe the term has been used, super genes. It's like a huge piece of the chromosome that's got all kinds of genes, and they're kind of, these chromosomes are kind of adapted to different environments, and the populations can like exchange them. So the whole block of genes that allows you to have earlier flowering time can be transferred like from one individual into the population, and the recombination rate is low so that it can all be included as a unit, a modular unit, and then it can spread through the population when it needs that to adapt rapidly. Okay, so the final bit of the research on herbarium genomics was on the leaf microbiome in invasive populations. So here we wanted to investigate those defense signals we saw in the selection of particular genes in the host over time. And we wanted to know if these, more about this rapid evolution of defense A little bit of early work from Vanessa, again, showed us that if you look at historic herbarium specimens, if you look at multiple species and which like different types of plant hosts, like Arabidopsis and ragweed, you find some of the same microbes present on just the historical specimens in comparison to the modern. So we view these as herbarium contamination. These are microbes that are living on the plant after it's killed and collected for the museum. And those are basically contamination that we don't want to include in our analysis. So Vanessa came up with a kind of a nice way to get that list of taxa and remove it from our analyses in order to get a more accurate picture of the taxa that were actually present on the plants when they were collected in the historical period. And that was an important part of the remaining part of the study. So the most recent work we're doing to look into the microbes on the plants has been involving metagenomic assembly. This is a really cool, a very advanced process that Jaya Labrilli, a postdoc from my group, is really good at. And basically what she's been doing is taking all the reads that don't map to the reference genome of the plant and using all of that shotgun sequencing data from Illumina to do de novo assembly. And in the perfect case, we end up with a complete circular genome of all the bacteria, for example, or a complete genome of all the fungi that are present in this mixed environmental sample from the surface of the leaf in this case. And you don't need a reference genome for that when it works. So the quick process here is that you take your reads that don't map, you do the de novo assembly using your specialized software, and you end up with contigs. And these contigs are binned by some software into what is thought to be like a single genome. So somehow it's able to separate the blue parts of one blue genome and put them into a bit like a circular genome. And to separate all of these into different taxa. And then they can be annotated but by what their closest relatives look like, and we can start to say something about the whole community based on the whole genomes of the microbes, not just conserved markers. When we do that kind of analysis on our 750 genomes, we quickly find that many of them are too low depth sequence to really do much for. So we do a huge amount of filtering to look at the ones that we can really trust. And here you see a PCA of the microbial communities from a bunch of specimens, including modern and historic Europe and North America. And what you should see here is there's not much of a signal in the PCA. That's what we found, not much structure. But we still wanted to know whether there was like a little bit of something going on in these microbe communities. So what we did was to look for if any microbes that we detect from our metagenomic assembly were changing a lot over time, either in Europe, from historic Europe to modern Europe, or changing in space from North America historical period to historical Europe. So that's basically taxa that became more common right after they were introduced to Europe. And when we do those two comparisons, we found like 150 significantly different microbes. And then we focused on the 33 that were strongly significantly different. And here you can see an example of this Ramularia microbe, which was kind of lower frequency in historical North America and then became higher frequency in historical Europe. That's interesting. Why did that happen? Well, that's what we wanted to do next. So we wanted to do a GWAS. We wanted to find if there was a statistical association between this change in frequency of microbe and particular variants in the host genomes. Okay, and this is kind of a new thing that is only just recently possible to do. And we wanted to do the same, like comparing the two comparisons, North America to Europe, and then over time in Europe from past to present. And we find, luckily, some interesting regions of the genome that are associated with the change in abundance of these microbes. Here we see this peak on chromosome 15, which is highly significant. And inside there, we found some genes related to defense function. And this particular fungal pathogen was first discovered in Europe in the 1800s. So that actually was thought to be introduced to Europe in the 1800s. So I think we might have actually just caught some of that signal here where the introduction happened. But it seemed to have affected the ragweed genome in a way. So I think here we see some selection, either probably against this pathogen. Okay, and there were a few examples of this. Mostly fungi that we found were changing in abundance, either in time and space, and that were associated with genes in the genome that kind of made sense. Again, this was a resistance to a pathogen. Here we had a bacteria where we actually got the entire genome of this bacteria recovered, and it used to be more common in the native range, and then in the introduced range, it's less common. So here, I think we're seeing the loss of selection on that microbe. And another case with defense. Okay, so we're getting into these results more, and in the future, I think what we believe is that some of these links to defense genes suggest that this is a real host microbe interaction. So I think we have actually found some kind of thing that's been going on in the evolution of the host in response to the environment with these microbes. And we'd like to know whether the variants behind these, the genes in the host have been highly selected. And we want to characterize that a bit better in the future. And then we want to know more about the ecological functions of these microbes, because basically all we've been doing is Googling their names so far. So we don't know much about them really. And for many of them, we may never know anything, but this can help us to understand better whether enemy release has been important in the evolution of invasion in common ragweed in Europe. Finally, I would like to conclude that herbarium specimens are a really rich and cool source of information for evolutionary genomics studies, because there's so many of these specimens and they're so well distributed in time and space because botanists have been so busy for the past 400 years. You can do these population genetic studies and never have to go to the field. Or in any case, if you like going to the field, you can compare your field collected specimens with specimens taken 100, 200, 300 years ago and learn a lot about that. And these approaches don't just apply to plant collections. Any museum collection, you can really do the same thing if there's enough specimens of your taxon of interest. Okay, I talked a long time, and I'd like to say thanks again for inviting me, and thanks to so many people who have been involved in the research that it is impossible to name them all. But in particular, Vanessa Beaker, this is much of her work, and Jaya Labreely, and Kay Hodgins, who's in Australia collaborating with me on this, and my research group here in Norway, including some Peruvians like Renato and Fatima, who are here for 2-year scholarships and master students here in our group, and we're very happy to have them. So thank you.